Hello everyone, thank you for the opportunity given by the organizer to present my work as a video presentation. My name is Muhammad. I am master student from Mondosmas University Department of Nanosciences and Nanotechnology, Samsun, Turkey. Today I am going to present a part of my research, which is the actinobacteria-related sciences of titanium dioxide nanoparticles and their potential as pesticides. Under the supervision of the Professor Mrs. Assistant Professor Dr. Hilal I. The plan of my presentation, which is small introduction of the context of my work, then the objectives of followed by material and methods used in this work, and achieve with the result discussion and the conclusion. Nanotechnology is the sciences that studies and the manipulation of the matter at the atomic and molecular scales. Nanotechnology is concerned with the creating new technologies and means whose dimensions are measured in the nanometers, which is the 1,000th of the micrometer, 1 million of the millimeter. Nanotechnology is the considered an essential aspect of the research field, various industries, including manufacturing, cosmetics, medicine, electronics, and agriculture. The continuous use of chemical pesticides has damaged the environment, negatively impacted agriculture production, and reduced agriculture sustainability. The use of synthetic pesticides has resulted in the development of resistance by the pest organism in the continuous exposure to the pesticides. Different approaches have been used to synthesize metallic nanoparticles as toxic chemicals used in the physical and chemical methods may remain in the nanoparticles format, which may prove hazards to the environment and organism in their application. Green synthesis is an efficient technique that includes synthesis through plants, bacteria, fungi, etc. Nanoparticles synthesis by bacteria may produce more catalytic activity and limit expensive and toxic chemicals. The plan of my presentation will be: first objective is to determine the antifungal activity of the synthesized titanium dioxide nanoparticle against plant pathogens, fungi, Fusarium, Glomorum, Fusarium, Graminarium, alternata, and Rhizoctonia cerealis. The second objective is the study to achieve the inhibitory activities of the titanium dioxide nanoparticles of the germination ability of radish Rhamnosus savis. Material and method: selective isolation of the actinobacteria. Soil samples were collected from 5 different cities in Turkey and Iraq. 1 gram of soil was taken in 9 mol of Ringer solution. Oxide soil suspensions were treated after treatment. And then 7.1 mol of each suspension was spread over actinobacteria isolation agar for 3 weeks. Actinobacteria-like colonies were selected and purified on glucose extract, yeast, malt extract agar, tryptone agar media, yeast extract, and glucose. Purified isolation were mutated in glucose stock solution at -20. Actinobacteria isolates were inflated into tryptone yeast extract glucose broth media and incubated for a week. Then broad cultures were centrifuged to synthesize titanium dioxide nanoparticles. The separated was added dropways with continuous sling into titanium oxysulfate TiOSO4 solution. Then the solution was heated in a water bath with shaking. After the formation of the white colored precipitated at the bottom of the flask, the titanium dioxide nanoparticles were centrifuged and washed with sterile distilled water. Then we got titanium dioxide nanoparticles. Characterization of titanium dioxide nanoparticles. Firstly, UV vis spectrophotometer, electron microscope, beam and seam X-ray diffraction. Results and discussion. Isolation and identification of actinobacteria. Out of 130 isolates, 10 isolates were found to synthesize titanium dioxide nanoparticles. These isolates were identified by a molecular methods based on pairwise comparison of 16 sRNA gene sequences, and the 9 isolates were determined as members of the genus Streptomyces, while one strain was found to belong to the genus Rhizococcus. The phylogenetic relationships of the isolates with the genus Streptomyces and Rhizococcus were analyzed by maximum likelihood and maximum permosome algorithms figure 2 and figure 3. Antifungal and phytotoxicity assays. The ability of the titanium dioxide nanoparticles synthesized by the actinobacterial isolates inhibit the growth of 4 plant pathogens fungi was tested. Comparing to the positive control known of the nanoparticle samples exhibited bioactivity against the 2 plant pathogens fungi tested in the present study. On the other hand, phytotoxicity of the titanium dioxide nanoparticles were analyzed for germination inhibitory activity for radish seeds, and the nanoparticle samples were highly effective too, with titanium dioxide nanoparticles could germinate while control seeds were germinated under the same incubation conditions. Physiochemical characterization of titanium dioxide nanoparticle. The UV spectra analysis performed one range of 200 to 900 nanometers wavelength showed that the titanium dioxide nanoparticles synthesized by the actinobacterial isolated exhibited the absorption peaks at 250 and 350 nanometers ranges due to the expansion of surface plasmon vibration. Futures physiochemical characterization based on X-ray diffraction and seam EDS analysis was conducted for the titanium dioxide nanoparticles synthesized by strain ESA 450. The XRD pattern revealed that the nanoparticles showed deficiency in distinctive diffraction peaks, which corresponded to amorphous nature of these nanoparticles. The morphological features of the titanium dioxide nanoparticles that derived from strain ESA 450 were characterized using SEAM. The observed particles are granular and morphoid cultures. The elemental analysis of the synthesis nanoparticles was carried out by EDX analysis. The energy displays XR spectrum of the titanium dioxide nanoparticles forming the presence of the titanium region in the ring of 0.6 kV through shape spectral singles. Moving to the conclusion, science of titanium dioxide nanoparticles using the green approach is a simple, inexpensive, and eco-friendly process that reduces toxicity chemicals. In the present study, the green synthesis of titanium dioxide nanoparticles was achieved using actinobacteria isolated from soil samples. The titanium dioxide nanoparticles showed strong inhibitory activity for radish seed germination. Therefore, titanium dioxide nanoparticles synthesized by actinobacteria can be formulated as herbicides for controlling weeds in agriculture fields. In addition, phylogenetic analysis of the actinobacteria used for nanoparticle synthesis revealed that several strains might be novel species in the genus Streptomyces. Consequently, further work is required to ascertain the taxonomic provenance of these strains. Thank you.